A very warm welcome and good afternoon to each and everyone present here for the international webinar on herbal alternatives and complementary medicine for COVID-19, organized by Department of Botany, D.D.U. Gorakhpur University, Gorakhpur. My name is Dr. Deepa Srivastava, and I have the privilege of being your host today for the webinar. Our webinar is a celebration of knowledge. I am super excited about the lectures we'll have today, as we have two amazing speakers who will share their expertise with us. Before we get started, let us have a huge round of applause for Almighty God, as with His will, we are here together. Now, I would like to start today's event by introducing our respected dignitaries. First of all, I heartily welcome our Honorable Vice Chancellor and Chairperson of today's event, Professor V.K. Singh. Thank you, sir, for your support. I am blessed to welcome Professor Hari Sharan, Pro Vice Chancellor, D.D.U. Gorakhpur University, Gorakhpur. Thank you, sir, for providing your precious time for being with us. I am extremely happy to welcome Professor S.N. Tiwari, Dean Faculty of Science. We thank you, sir, for being here. We welcome you, sir. It is my privilege to invite our chief guest of today's event. So, with a warm round of applause, please welcome Dr. Badri Narayan Pandey, Scientific Officer H, Head Radiation Signaling and Cancer Biology Section, Bhabha Atomic Research Center, Trombay, Mumbai, India. He will be delivering his talk on potential of low-dose radiation therapy in management of COVID-19 patients. Thank you, sir, for gracing the occasion. It is my pleasure to invite our guest of honor, Dr. Vaidurya Pratap Sahi, a very young and dynamic scientist at Karlsruhe Institute of Technology, Germany. He will deliver his talk on botanical globalization, Tulsi and Moringa. We are happy to have you here, sir. We welcome you, sir. Now, I invite Professor Malvika Srivastava, Senior Most Professor of Department of Botany. We welcome you, madam. It is my privilege to invite Professor Ishwar Das, sir, Emeritus Scientist, NCSTC DST Program, and ex-head Department of Chemistry. Thank you, sir, for gracing the event. I am blessed to welcome Professor V.N. Pandey, convener of this event, and head Department of Botany, D.D.U. Gorakhpur University, Gorakhpur. We are thankful to you, sir, for organizing this event. Now, I welcome Professor Pooja Singh, coordinator of the program. Thank you, madam, for being with us. May I especially welcome Professor A.K. Dwivedi, co-coordinator of the program. Thank you, sir, for being a part of this event. Now, I welcome organizing secretaries, Dr. Tulika Mishra and Dr. Rakesh Pandey, who have been the key people in helping throughout to design and take this webinar to the stage that it has become the reality of the day. We welcome you both. Now, I welcome all the eminent scholars and honored guests from BAC Mumbai who have been so excited to become part of this event. I welcome all the coordinators, head, dean of various departments, faculty members, students, and the esteemed audience who have gathered here from the diverse places across the country and from abroad. Again, on behalf of Department of Botany, I welcome all of you for being part of this event. Thank you to each and everyone for being here today with us. Now, I would like to take you through the purpose behind today's webinar. As we all know, this year we are facing global pandemic as COVID-19. Our day-to-day life has totally changed as the cases are increasing in India, including Gorakhpur. We need some botanicals for our purpose, for boosting our immunity. We should also know about some alternative approach which may be possible to provide us rays of hope. So, for this, we have with us our renowned scientists to guide us and our student in this stressful condition. I hope that all of you will have 2 hours of most productive and stimulating discussion. I really wish that this webinar will be a great success, not only as an opportunity to share knowledge and experience in higher education, but also a beginning of a long and fruitful cooperation and friendship among fellow educators devoted to most meaningful and worthwhile tasks and teaching and training our youth who will shape our future. Now I request Professor S.N. Tiwari, Dean Faculty of Science, to bless us with his message. We request you, sir, Professor S.N. Tiwari.
Thank you, Dr. Deepa. Are you listening me?
Yes, sir. We are listening you, sir.
First of all, I would like to thank all the faculty members of the Department of for very active involvement in academic activities throughout this pandemic period. And this is one of the most important organized by the Department of Botany, an international webinar on and complementary medicine for COVID-19. On this occasion, I would like appreciate the cooperation and help extended by the advisors, Professor Ishwar Das, ex-head and emeritus scientist, NCSPC DST program, and Professor Malvika Srivastava, head of the Department of Microbiology, the coordinator of the program, convener of the program, Professor Vasisht Narayan Pandey, and co-coordinator, Professor Anil Kumar Dwivedi, and organizing secretaries, Dr. Deepa Srivastava, Dr. Rakesh Pandey, Dr. Tulika Mishra, and other faculty members of the Department of Botany. And I'm least hesitant to say that the Department of Botany of our university, and particularly of our Faculty of Science, is the most academic department, and it has been continuously organizing several programs throughout this lockdown and shared very important information regarding this, regarding the management and cure of the COVID-19 virus. I'm very much thankful to our chief guests of this occasion, Dr. Badrinarayan Pandey from Bhabha Atomic Research Center, Trambay, who is the speaker, the learned speaker of this session, and another guest who is connected with us from Germany, Dr. Baidyur Pratap Sahi. These two learned speakers are going to illuminate us how to manage the COVID-19 pandemic. And the first lecture which is going to be delivered probably is the management of COVID-19 through the radiation dose. We are all exposed with radiations every moment because we know that every particle, every system, every object, whatever it is, that is continuously radiating and also continuously absorbing radiation. So radiation effects on materials and interaction are, in other words, we can that is more popular. Interaction of radiation with matter is occupied the most important area in the present day research. Because if you want to grow your crop, if you want your product for a longer duration, if you want to cure even cancer patients, whatever uh, you like to do in material science or in health sector or in medicine or in agriculture, all are affected by radiation. So I am a firm opinion that radiation play a very important role in our daily life, and every moment is not that important where you are. We are living in a closed AC room, even then radiations are important for us and full managed. But something we don't care that we never bother about the water, air, because that is available to us easily. Like that, we never bother about the radiation. But those persons who are working in the field, particularly coal mines or in radiative stations, atomic power stations, are people who are working with nanomaterials or in nanotechnology. They also use radiation. Those researchers are always bothered about radiation. And undoubtedly, we all know that radiation therapy has emerged as one of the most important sectors of the cancer therapy, not only in the area of cancer therapy, but in other several areas also. Not going into much detail, we are eager to hear learned speakers as expertise in field. But as a student of physics, I must say that this is going to be a very important and informative lecture because ionizing radiation having energy more than 10 electron volts. These are called ionizing radiation. So we are going to have a lecture on ionizing radiation in the cure of the COVID-19. So I once again extend my warm welcome to the chief guest, learned speakers of this webinar, and extend my salute to all the members of the organizing committee and the Department of Botany who has been continuously engaged in so many activities throughout this pandemic period. With these words, I would like to also extend my courtesy to to our Vice Chancellor, sir, who has allowed us to hold such type of activities throughout this pandemic period. With these words, I once again welcome the chief guests, learned speakers of this session, and the people involved from the Department of Botany in this activity. Thank you.
Thank you,
sir.
sir. Thank you, sir, for boosting our morale. Thank you so much, sir, for such a nice speech. Now I request Professor V.N. Pandey, Head of the Department of Botany and convener of the program, to give light on the theme of international webinar on herbal alternatives and complementary medicine for COVID-19, organized by Department of Botany, D.D.U. Gorakhpur University. We welcome you, sir.
Good afternoon, everybody. I welcome all of you in this international webinar from my core of art, and on behalf of Department of Botany, faculty members, organizing secretaries, my dean, Professor Subhriv Nath Tiwari sir, BBC Professor Harisharan, and our Vice Chancellor, Honorable Vice Chancellor Professor V.K. Singh. They have given strength to arrange these types of webinar in the present problem and scenario of the country and the world. Now, presently we are facing with the COVID-19, how plants are valuable to overcome the problem of COVID-19 as a alternative medicine and as a complementary medicine. Many spices and many herbal medicines are available in nature. They have more strength and power to remove and to reduce the adversity of the COVID-19 virus, as well as many complementary food and many complementary medicines are also available in the nature and the natural system to reduce or to cope up the problem of COVID-19. Both alternative and complementary medicine from herbal sources available in and around the system to maintain the strength and immunity of our body. When we have our full strength, full power in the body, and immunity through the plants available in the nature. Many spices available in the nature. For example, pepper, cardamom, black pepper, dry ginger, haldi, turmeric. These are available in the nature, and when we are utilizing in more or less amount in day-to-day life, its dose is important, and that dose is required in our day-to-day life to maintain the strength and immunity of our body. In another way, we have heard about the complementary medicine. Complementary medicine are those medicine which are available in the nature, just like a protein. Proteins are building blocks of our body day to day in our life as a food. But when children are suffering from kwashiorkor, marasmus, mental retardedness, keratomalacia. These proteins are used as a medicine, and that medicine at that time regarded as complementary medicine for the children. In the same way, this is also important. Complementary and alternative medicine are also important for mother suffering from many diseases, or lactating mother, or pregnant women. They also require amount of proteins and energy, so they can be given in a sumptuous amount to cope up the problem of COVID-19 or viruses that affect us. So in this way, we have two learned speakers from one from our country, Dr. V.N. Pandey, and another from Vaidur Pratap Sahi. They are competent, having a great expertise to enlighten us how to overcome the problem of COVID-19 in India and abroad. And many of participants from different part of country are also associated and seen on the list. Dr. Vinay Chaube ji, Rohit Sharma from BARC. I welcome all the participants in this way. Now, one of my advisor, Dr. Ishwar Das, is eagerly. Here is a physics, Professor Sukhdev Nath ji, chemistry, Professor Ishwar Das ji, and radiation biology, radiation biology, and Vaidur Pratap Sahi from Germany. And we are the plants representatives, living with the plants on this planet with the people of country and people of the world. So I shift my mic to the Dr. Deepa Srivastava to request our learned speaker, Dr. Deepa ji.
Thank you, sir. Thank you, sir, for such a nice introduction. Now, I request Dr. Rakesh to start technical session 1 and introduce and invite our guest, Dr. Rakesh.
Thank you, ma'am. Very good afternoon to all of you. In this technical session 1 of this international webinar, we have a very eminent, dynamic, energetic, and a humble personality with us, our chief guest, Dr. V.N. Pandey, sir. He is Scientific Officer H in BARC, also head Radiation Signaling and Cancer Biology section in Radiation Biology and Health Sciences Division, Bhabha Atomic Research Center, BARC, that is situated in Bombay, Mumbai. Before inviting sir for his lecture, I would like to share some words for him. Sir has expertise in radiation biology, cancer biology, cell signaling, and in cell culture. His team, under his guidance, recently has developed a thorium detection assay in the biological system. Sir has also worked on the hyperthermia therapy of cancer. Sir has also expertise in receptor tyrosine kinase signaling in cancer radiotherapy. Sir has expertise also developed in genomic instability in cells exposed to high and low radiation energy. Sir has published more than 130 research papers and has a member of and he is a member of many scientific societies. His publications also got citation over 2200. He was also a counselor in Asian Association of Radiation Research during 2013 to 2017. He was also a fellow of UICC, that is the Union of International Cancer Control Society, UICC Switzerland. He has run several research projects and member of various scientific society. So without taking much time, I feel immense pleasure to invite sir for his lecture. I request sir to deliver lecture. Thank you, sir. So now, sir, it's time to question and answer. And your informative lecture uh, provoke many questions in young minds, and I would like to take some of them. So the first question is, sir, does this therapy has any adverse effect on our immune system? This question is from Vandana Yadav, MSc 4th semester student of our university. Does this therapy has any adverse effect on our immune system?
Actually, that device I showed in one of these slides. Actually, in fact, it is not having adverse effect, but in fact, it's supposed to activate our immune system. It is like immune booster. What we use different types of immune boosters. So this, since it is very low dose of radiation, so it is sort of an insult. We are challenging our body. So our immune system get activated. So they are not going to have supposed to be any adverse effect of this therapy, especially our immune system. And even some of the cases, some of the blood cells may go down, but ultimately it will recover fast. I hope I'm clear.
Okay, sir. Okay, sir. Another question is from Arema Mishra. Can radiotherapy be applied with other therapy simultaneously, or it acts in a synergistic way?
Actually, not much known in case of COVID, and that is why I showed in one of the slides that there is a few days back one paper came where they here discuss about that it can be used for convalescent plasma. But still, like since the whole low dose radiation therapy itself is a very new area, and even COVID itself, like not much research, clinical research and more, but a lot of things can be tried. At this stage, only one paper, to my knowledge, it came where they talked about the combining the convalescent plasma with the low dose radiation therapy.
Okay, sir. Okay, sir. This is another question from Neha Pandey. If this therapy does not remove the virus completely, what are the chances of having infection again once the therapy is done?
Actually, this therapy is basically not directly killing the virus. That I wanted to make you very clear to the audience. So if you wanted to kill directly with radiation to virus, it requires a lot of dose, and that patient cannot survive. This is basically indirect killing of the viruses. So if you are killing by any mode of this one, so whatever chance is there of recovery again through the virus coming up by the virus the patients, it is going to be same in case of here also. But here, since it is a more indirect way of killing the virus by immune activation, so I feel that they are going to be very less likely to again coming of these viruses again to the patients.
Okay, sir. Thank you, sir. Another question is from Ananya Singh. Cancer patients undergo many therapies, yet they are prone to COVID infection. Why?
Cancer patients go to many therapy?
Sir, okay. Cancer patients undergo many therapies, radiation therapy, yet they are prone to COVID infection. Why?
Since actually one thing I wanted to make to the clear to the cancer patients are already having very low immune system, very poor immune system due to the cancer. Since cancer is also basically immunosuppressive disease. So they itself cause the suppression of the immune. So already patients are having lower immune system, and actually person is having lung cancer, etc. So they are more likely to develop the cancer. This is one of the way, one of the cause of cancer patients are having more chances of developing COVID infection.
Okay, sir. Sir, another question. In some of the virus-infected patients, it has been observed that blood clots are formed in whole body. So can this radiation therapy remove these clots also, or this only confines to lung inflammation?
Actually, till now, this low dose radiation is mainly it is advocated to cause more anti-inflammatory pathways. So what happens that if they are able to survive the clot are formed, their clot are formed due to the viruses are trying to aggregate this RBCs actually, and some of the spike, etc. So those things are there. So if you are able to eliminate the viruses, we are certainly, I feel that we are also going to eliminate the possibility of developing the clots and causing the cardiac problems. But still, not much research is known in the low dose radiation therapy. That is what this is still like, this is a gray area. And this is what, like, whatever till known, actually, to my knowledge, I try to present most of the evidence or most of the literature.
Okay,
But
sir.
certainly, indirectly, it will cause the clot formation, but not directly, I feel.
Okay, sir. Thank you. Another question is from Mr. Saurabh Anand, ICER Pune. Sir, can we use this method for large number of people as it may be costly?
No, actually, see, large number of people, since now the advantage of this system is that already we have cancer hospitals available. And now cancer hospitals either use for, these are the basically radiation is coming out from them. Only you need a person who is going to treat it. So now if you can treat, most of the cancer hospitals that operated for maybe few hours. If you want to have more patients, you can operate for overnight. And as I told you, that does not require very long time as well. Like, since we are going to use very low dose of radiation, hardly we are less than a minute we can treat it. So I don't think it is going to have a lot of problems for the treatment in terms of, and the most advantage that already for lung cancer patients, you have already got treatment planning, everything known. Only instead of cancer patients, you have to place the COVID patients. So that way it is, I feel that is going to be very useful and it can be explored.
Okay, thank you, sir. So we wish that this radiation biology and radiation therapy will be helpful for combating COVID-19 pandemic and many more diseases. Thank you once again for your very well informed lecture. And now I request Dr. Deepa to continue the session.
Thank you
Dr.
very
Deepa.
much.
Thank you, sir.
Thank you, Dr. Rakesh, and thank you, sir, for such a nice lecture. Now I request Dr. Tulika to start technical session 2 and introduce our guest of honor, Dr. Tulika.
Hello, am I audible, Dr. Deepa? Hello?
Yes, you are
Hello.
audible.
Yes, ma'am.
Okay. Thank you. Due to some connectivity issue, I have closed my video, but I welcome all the speakers who are here, and I also thank Professor B.N. Pandey from PAR for a great lecture he has given. Now we will move on to our next speaker, and we are very lucky to have Vaidure Pratap Sahi between us. He is from Karlsruhe Institute of Technology, Germany, and he is going to deliver a lecture on the topic Botanical Globalization, Tulsi and Moringa. This university, which Sir belongs and is doing his research there, is a great university of Germany, a very leading university, which is known for its inventors and entrepreneurs. As of 2018, 6 Nobel laureates are affiliated with KIT, including Heinrich Hertz and Carl Friedrich Benz. So we are so much glad that you are with us, sir, and I will like you to give your lecture and embrace us. Thank you. Dr. Vaidure Pratap Sahi.
Hello,
Hello, yeah.
can you hear me?
Yeah,
Yes,
we can hear you. Yeah.
Okay, and just and is my slide visible?
No, it is showing that you have started screen sharing. It is yet not visible.
So it will start in few minutes, few seconds.
It will start in a few minutes, sir. Till then, can you make us more acquainted with yourself and
Hello?
what is your work there? Hello.
Can you see my slides?
It will be open in just a minute. It will be opening. Till then, you can just start and tell about your work.
I can't hear anyone.
Sir, you are audible.
Hello?
Yes, you're audible now. The screen is not there, but you are audible.
Hello.
Yes, sir.
I'm very sorry, but I cannot share my slides, and I don't know what's going on because it just states that Dr. VP Sai has started screen sharing, but I cannot see my screen on the webinar.
Yeah.
We are trying, sir.
Okay, just a moment.
Hello,
Hello.
sir. Sir, can you mail your PPT? Sir, can you mail your PPT to us? Hello, hello.
हेलो राकेश जी,
मैडम सर से आप कहिए कि वो अपना पीपीटी मेल कर दें।
Sir, please send your slides to me. Shahi sir, are you listening? Please send your
I
slide to
cannot,
me.
Dr. Deepa, I cannot hear you actually. I can see your
Hear me?
video, but
Please send your slide to me. He is saying that he is not hearing me.
Hello. 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 Hello.
Hello?
Hello. Yes.
Hello,
Is it
sir, we can hear you. Sir, we can hear you.
is it now visible?
Yes, sir, some KIT disclaimer. Yes, yes, it is quite visible. Okay, sir.
Okay. Great.
Yes, yes, your mobile is visible. Mobile screen is visible.
So I can start now.
Yes, sir, yes, sir, yes, sir. Thank you, sir. Yes, sir, you can start.
Okay. So I'm sorry everyone for the confusion and the problems, and I would like to thank the organizers for having me as a
Yes,
speaker.
sir.
So am I audible?
Yes, sir, you are audible.
So very good afternoon to all of you. And since we are in a time where we have learned in this time of Corona how to communicate efficiently using the online mode. So it's a good opportunity for me sitting here in Germany to talk to all of you from D.D.U Gorakhpur University, because I have a very personal connection with this university, not because I studied there or I was a part of the alumni, but because of the foundation of this university has a strong connection with my family. And can you see this picture?
Yes, yes, yes.
There's an old picture here. Is it visible?
Yes, yes, it is visible.
This is one of the first few pictures of the foundation of the Gorakhpur University, and in the picture below you can see Dr. S.N.M. Tripathi, Sir Majithia, and Mahant Digvijaynath from the Gorakhnath temple. So they were good friends of my great grandfather, and who was Raja Indrajit Pratap Sahi of Tamkuri, and that's how I have a personal connection with this university. So it's a great opportunity and a very big honor for me to be talking with faculty and students of Deen-Dayal Upadhyay University from here in Germany. So thank you very much. Thanks to the Vice Chancellor, thanks to the Professor Pandey and the organizers. Thanks to Dr. Deepa Srivastava for having me here. So I'll start my presentation. Just keep telling me if I'm audible. Hello?
Sir. Yes, sir, we can hear you.
Okay, great. So the thing is that we have learned during. So one thing is for sure that there has been no cure for corona, or there's been no vaccine for corona. But one thing is there that we cannot cure the COVID-19 as of now, because we are all everyone I mean scientists all around the globe are working on it to either find a vaccine for it or to find some proper cure for it. So it's too early to think of finding a cure or a proper vaccine for it. But there's one thing which we can work on, and this needs a long-term um, process, that is by building habits by which we can live uh, healthily, in a more healthy manner, so that we avoid certain diseases to come to us. So this is also one of the ways, one of the sustainable goals of the food and agriculture organizations, that is to develop smart foods. Now, what is a smart food? So if you ask me what is a smart food, it is like saying, okay, it doesn't have to be intelligent to be smart food. Smart food is something that if you take a small amount of it, you get enough nutrition, you get enough, some added values from that food. It has some therapeutic properties, or it is rich in certain nutrients. So once there, at least I have been here in Germany for the past 4 years, and in Europe for past like 7 years. So I have seen a trend in the market for these smart foods, and one of the smart foods which has a very common place is moringa. So can I hear, can all of you hear me, please? Hello.
Yes, sir. Please continue.
Yeah, so I will be introducing you to a plant called moringa, which in Hindi we call sahjan. I mean, we all know. So there's a nice anecdote about it. In my own life, in my own scientific career. So when I came to Germany, I did my PhD in Japan, and then I was a postdoc in Prague, in Czech Republic for 3 years, and then I moved to Germany. So we have in our lab every Monday a seminar. So students come and present. So I was sitting in the seminar, and the topic of the presentation was moringa, and here in Germany it is considered a superfood. So I was like, oh, moringa, sahjan. We all use sahjan in sambar, or as a sabji, but it's not so. I mean, we never thought that. We never think. I mean, many of us, at least I did not, when I was a child, and I did not like moringa very much. So I was like, what is so super about it? Then we were discussing, and then I called home. My native village is Tamkuhi Raj in Kushinagar district. So I called home and I asked, and then I was talking to one of the. I was like, there's a person who works on our farm. I was talking to him and I said, how do you use sahjan? So in Bhojpuri he said, हमनी के गइयो सहजन के पता ही ना खाली सन. So whereby he meant that even our cows do not like to eat the moringa leaves. And I was like, okay, even our cows are not eating, and here it's like 100 grams of it is selling for 16 euros. So there must be something interesting in it. We started looking at it, and I will just give a brief introduction to it. So it comes from the order Brassicales, and it's from the family Moringaceae, and the genus is Moringa. So there are many types of Moringa species. Many species of Moringa are found in the world, and most of it is restricted to Africa and Asia. And there are two species which are from India. There's one from the Middle East, and the rest of them are from Africa. So it's a very strange. It has a strange distribution, or rather not so strange, because we all know that from the Gondwana times it might have had a common ancestor. So yes. So now a little bit about the etymology of moringa. So moringa, as I already said, in Hindi it's nothing else but sahjan, and here is a distribution of the moringa uh, species, different species. So as I said, so moringa oleifera, which we see commonly in our uh, country in India, and the other one is moringa concanensis, which is also found in southern India and parts of western India. And then there is the moringa peregrina, which is found in the Middle East, in the Arab Peninsula. And then there are moringa stenopetala, ovalifolia, longituba, etc., which are found in Africa. And then there are two very special moringas, the moringa drohardi and hilberdranti, which is found in on the Madagascar islands. So as you can see here, if you look at the phylogenetic tree below, you see that there are different types. They are clustered in different the species are clustered based also not just on their genetic orientation but genetic makeup, but also on the basis of their anatomical features, like the different tree forms, for example. Now superfood, coming back to superfood. So uh, people started thinking here in Europe, like, and across the world, like, what is so special about it. And then when they started making reviews about it, and started to look into the matter more deeply, and they found that they did a proper analysis of the different parts of the moringa, and they found that if the flowers, the leaves, the pods, the seeds, and even the roots and the barks are used for medicine and are used um, for therapeutic reasons. They have functional properties. So it became it's called a super baum. Baum is a tree in German. So it's a super tree in many circles. So now, for example, you will be surprised that it has, like, generally uh, we know that moringa has I mean, spinach is rich in iron, but look here, it has moringa leaves have like that. There are reports which say that it has 25 times more iron than spinach, or 17 times more calcium than milk. So isn't this amazing? I mean, this is something which is like commonly growing in our backyards, but we are not aware of it. So this has a lot of potential for the local um, economy, and also it needs a lot of research on part of us scientists to understand the importance of this plant, and to sustainably utilize the potential which this plant species has for medicinal and therapeutic purposes. So I'm here sharing a very old book which I have. So you can see that I mean, it is very well defined in the Ayurveda, in Shushrut and Charak Samhita, about the importance of moringa. But I've just taken 3 examples here. So as you can see that it has therapeutic uses, as in cold, cough, skin diseases, and also the moringa flower has particular uses, especially when it comes to things related to the stomach, or even for the nervous system. And moringa has a special moringa flowers. The extracts of moringa flowers have a special significance in terms of eye infections, especially conjunctivitis. And a very nice thing, because we are talking about today, it's a topic of COVID-19 and Corona. So there's a very nice observation. This, like, we have this smallpox and chickenpox, or the measles. So monkeys also have that, and when they they feel that they are having this kind of a viral disease, they go and eat moringa leaves. So it might, I'm not saying that it has, but it might have an antiviral, or it might have a use to have therapeutic uses against viral diseases. And that's where it is important to study this plant in more detail and in a more scientific manner. So that was just from the Ayurveda, and not just that. So if you can look here, this is a very old book on elephants, and here the moringa root, the moringa flowers, and the moringa leaves have a mention in against uh, elephants, I mean against diseases of elephants. So for example, the bark and the roots are, they can be used when to relieve pains and also rheumatoid pains in elephants, and also they are very good in curing blisters. The moringa leaves, the powder from the moringa leaves are used for blisters. So this is like this really shows that it has potential not just in humans but also animals. And again, I am sharing um, a book from uh, a book on horse husbandry, and here again the bark of moringa tree is recommended as a powder for treatment of cold. Now you can well connect. We are talking about COVID-19 here, about corona, and here we have usages of moringa in treatment of animals or even for humans in terms of cold and cough, and certain diseases which have viral roots, so this is a plant which has a lot of potential to be studied and to be utilized for therapeutic purposes. And again, I stress on this, that this is not something like ashwagandha or some plant which needs a lot of inputs. It's a plant which grows in our backyards. It can be easily accessed, it can be easily utilized. So for example, in the European Union, the dairy farmers or farmers who deal with animals, they use special moringa powders for feeding the cattle, and it has given wonderful results in terms of milk and prevention of diseases like especially in cows and goats. Now coming to humans, so if you look at, can can you see the slide here? Can you hear me? Hello?
Yes,
Yes,
sir.
sir,
Yes,
we
sir.
can hear
Please
you.
continue.
Yeah, so here you can see these different moringa products, and you won't believe the moringa which a person in my village said that his cow does not eat, sells for a very expensive price, that is like 100 grams sometimes it's for 16 euros, and that's like, I mean, it is just unimaginable for a person who comes from a village from India. So the Europeans are utilizing it, and it's time that we also do it. So a little bit about the nutritional composition and the different medicinal uses of different parts of moringa. So for example, if you look at here, I mean, I can share this paper later with you so that you can have a look at it. So each part of the plant, like the leaves, or the flowers, or the roots, everything has an important, you know has an importance. So I'll later share these papers which I'm using in this slide with Dr. Deepa, and she can share it with you for your information. Now the problem is, yes, with good things come bad things. So and the bad thing is that with a potential of such therapeutic usage, moringa has a threat of adulteration. So it is important, that's what we are doing in our lab. We are trying to authenticate and use molecular markers and microscopical analysis, and that's what I will show you in the next few slides. So Goethe is a very good famous philosopher from Germany, and he said the hardest thing to see is what is in front of your eyes. So that's how we get cheated. We are told that it's moringa, but it's actually not moringa. And to control that, we are developing markers and techniques by which we can authenticate we can say about the authenticity of our products. So for example, here we did leaf characterization of Moringa oleifera, which is a very common Indian species. Then there's Moringa stenopetala, which is from Africa, and Moringa drohardi, which is also from Africa. And as you can see here, all three have different leaf patterns, and this can be helpful. But at the same time, and here we have a slide which shows the measurements, and you can see that, for example, the here we are showing the ratio of the breadth and the length of the leaves, and olifera lies between Drohadi and Stenopetala. But the problem is we do not have whole leaves when we have powders, and we have small pieces. So what could we cannot measure. So the next step was to look at the to look at microscopical pictures which were taken of the pavement cells, that is the epidermis of the leaves, and we did it for different species. And as you can see in panel D, there are some shining crystals. These are oxalate crystals which are found in the moringa leaves. So the same thing is with moringa stenopetala, and also with moringa drohadi. So in our, I mean, we did an analysis for different species, and in this table, in the extreme right hand side, you can see on the top two commercial products, that is herbal tea and this is bio moringa tea. So what it was very difficult to differentiate and to find out, because if you remember the previous two slides which I showed, the leaves look more or less the same. I mean, there are not differences. So microscopical analysis or just observing them macroscopically does not help. So, and also you can see here we took two uh, uh, from different plants of different ages. Moringa oleifera is the same species, but look, there's a difference. So I mean this is not a good technique to characterize or to authenticate the product. So we thought of a way of going for molecular authentication. And for molecular authentication, I mean, all of you know that there are different ways in which it can be done. One is through the genomic DNA, which is from the nucleus. The other is from using mitochondrial DNA, and then there's the chloroplast DNA. So we have used the PSB-tRNA marker from the chloroplast, and also you will see in the next few slides how we have tried to work on that. So uh, yeah, so as you can see in the first column, we have the different primers, and we use PSP-tRNH, we use ITS, we use YCF1, but ultimately it was the arms-based method of arms primers of which we selected from the PSP-tRNH sequence, which helped us to differentiate between um, for example, moringas from Africa and moringas from Asia. So, and this similar thing happened when we tried to use the arms for the YCF1, and this was able to, with this we were able to differentiate between oleifera, uh, which is our Indian moringa, versus all the rest of the moringa. So this is a like a real um useful way, and where one could be very much sure. So for example, if you look at this gel picture, you can clearly see that moringa oleifera and moringa peregrina, uh, they show a diagnostic band of about 300 to 350 base pairs, and whereas moringa stenopetala and hildebrandt and rauhardi, which are all three of them are African moringas, they have the band is at 550 base pairs. So this clearly shows a difference in the band pattern, and this helps in differentiation. So in conclusion, for this authentication, and another thing about authentication is the microscopic authentication. It might be useful, but this molecular authentication is like a is a nail in the coffin. Like it's the last thing which you can, it's the last word you can use it for sure, that okay, for sure this is Moringa oleifera and not Moringa stenopetala. So the difference between the morphological and molecular authentication is that, of course, morphological authentication is easy. It it, it is like not so expensive, but the problem is that it takes a little bit of time, and the accuracy, the main problem is with the accuracy. Whereas in molecular authentication, although it is not easy and it's expensive, but the accuracy is high and it's a fast method. So these are the students who work with me. So probably many of you know about this cartoon called Popeye. So he uses spinach. We have forced him to use moringa. And I would like to, there's another aspect which I would like to touch, and I would like to use Mahatma Gandhi's quotation that the earth provides enough to satisfy every man's need, but not for every man's greed. And if you look at this, these two maps, the global maps, so look at the picture where the malnutrition is most prevalent, and look at where moringa thrives. That the region where moringa grows exactly overlaps the region where malnutrition is most prevalent, and the regions where malnutrition is not prevalent is the region where moringa sells the most, that is Europe and America. So this is something which we have to think, and I mean, this is towards a holistic approach. So one has to you know think about this, the social and the economical aspect. We as botanists have to have a job for society, a responsibility towards society to bring such aspects out. So with this I would like to close my talk, and uh, I will take questions now if there are any. Hello, I cannot hear.
Hello? Yeah, yeah, sir.
So I was supposed to give also a brief talk on Tulsi, but I will keep it for some other time because I think I have a time crash because the I want to keep up with the time. So I will keep it for some another time and stop it with Moringa. So I'm open for
But if you have time, you can complete Tulsi also, sir. Because due to technicality, we could not hear initially. But if you can, there is absolutely fine with us. If can keep it, we can elaborate on Tulsi also. Hello. Hello.
Can you hear me? Hello?
Yeah. Yes. Yes. We can hear you.
Yeah.
Hello.
Yeah.
Yeah, we
I
can
will.
hear you.
I,
Yeah.
I have selected a few slides so that I can quickly go through the Tulsi
Okay.
thing also. So
Yeah.
yes. So Ocimum sanctum, which is now called as Ocimum tenuiflorum, is our Tulsi G, our
Yeah.
Tulsi.
Yeah.
So we I mean, India has, we all know about the relevance of Tulsi ji. We have Tulsi Vivah in our culture. It
Yeah.
finds a very important place, especially for people. I mean, for Hindus, it's of course very important, but at the same time for Jains and Buddhists also, it has a special reverence on a religious basis. But apart from that, it is more important in our lifestyle, in our holistic living. It has a special place because, for example, we all have, whenever we get cold or when we have problems, tulsi ka kadha is the most easiest way. Like, you won't believe, in Germany, when I have a 6-year-old son, and when we take him to the doctor says give him ginger and tulsi tea. So even German doctors are prescribing tulsi for, for developing immunity and for, coping with cough and cold and such symptoms. So that's the importance of Tulsi. So Tulsi is really globalized. I mean, when we talk of globalization, we generally talk of things like, IT and all these things, but we forget that long before globalization was happening in the ancient times, with plants and food and botanical globalization is very old. So, and Tulsi is one of those things which is like, which is one of the important products which India has been exporting since a long time. So, coming back to 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 the aspects which we are working here. So what the again the problem here is that, we have Ram Tulsi, we have Krishna Tulsi, we have one Tulsi, we have so many types of different Tulsis. But the problem which exists in the or which used to exist was proper identification of these different cultivars or different subspecies. So we know that different subspecies might have different chemical composition, which might have a different effect. Like, for example, basil, the Osimum basilicum, which we
Yeah.
use on pizzas,
Mm.
it is not tulsi, but it's
Yeah.
also basil, and it's also basil, and the common name could be
Mm.
called as tulsi. So, but it might not have the same effect as a normal thing. So, the idea was in order to authenticate. So, and it's really tough to authenticate because when you look at the NCBI database, it's full of full of sequences which are not correct actually. And when we try to contact these people, it was like not so easy. So we had a tough time, but we had to do the job. So what we did was, we used different. So this paper is already published in PLOS One in 2018. So we use this RBCL and MATCA and TRNF markers to also using PSB TRNH, and we did a kind of cluster analysis, and we found 4 different haplotypes based on the sequences. Yeah. So uh, now what was uh, what was the real result of our topic was that we found that Tenifolium and Gratissimum and were like the 2 main clusters which were separate, and then the rest of the basilicums were forming a completely different cluster. So now, we wanted to do, as I said, that it's the medicinal properties or the therapeutic properties or the volatile compounds which we smell. They all are because of chemical profiles, and each species has a different chemical profile. So, for example, here we are showing 2 pictures of flavonoids and essential oils, and you can very clearly see the difference in the pattern uh, of these um, components or compounds. And it was like, for us it was very important to show that not just the genetic makeup but also the chemical profile, because generally when we talk of speciation, when we talk of species, we are restricted, or we restrict ourselves to the sequences or the genomic DNA, or on a sequence level, but we sometimes tend to forget that there can be a chemical profile, there can be anatomical features, so we have to consider that. So that's why we did this, and we could see a clear difference in the chemical profile of the species, and this was important. So I don't know why, but my next two slides are not working. So I would like to, I mean, anyhow, I'll continue because of some technical issues. I cannot share my slide properly. Just a moment. No, it's few of my slides are not showing in this particular instrument which I'm using right now. Nonetheless, I'll continue. So
Okay.
we developed, as I showed in the gel data of moringa, we have developed markers which clearly can differentiate between tulsi, I mean the real tulsi ji which we have, the sham and krish tulsi which we all have in our homes, and the other ocimum species. So which is very remarkable, because then if, for example, if I go to the market and buy tulsi tea, and if I have a doubt, I can clearly go to a lab and say that please do this test, and this is all open access, so you can always use this, and this is one aspect which in which I think as a botany department you can continue to work on, because this is something which is lacking in India, because we do not have proper authentication, molecular authentication techniques to identify Ayurvedic medicines. So apart from the, on one hand we have to still move on to authenticate our Ayurvedic practices, but at the same time we have to work to authenticate the products also, because there's lot of discrepancies in the market, and this sometimes can lead to problems. So with using these two examples of moringa and tulsi, I just wanted to show how globalization occurred on a botanical level, and how along with this globalization, Ayurveda was in the picture, and how it has helped to bring Ayurveda to Europe or to the West, and at the same time how important it is to validate these practices through clinical trials, through I mean for the practices through clinical trials, and of course for the products through things like what I have just talked about. Thank you.
Thank
I'm
you,
open
sir.
for questions now.
Yeah, thank you. Thank you, sir, for delivering such a nice
And
lecture.
I must say, I must apologize because of these technical
Uh,
errors. I was I had a lot to talk, but it just took up a lot of time for this because of this technical problem. But nonetheless, it's fine.
yeah, I finally we said "Ant bhala to sab bhala." So
Huh,
finally,
exactly.
did connect, and we got this opportunity of seeing your slides and hearing to you. And your research is very nice. And in this wake of COVID-19, as I would say, that the immunity boosting plants have taken the first seat or the driving seat in our research. And now we are seeing so many immunity boosting plants. And the basis of our research is definitely the traditional one. And as you said that we have a lot of Ayurvedic preparations and concoctions are there, but definitely the molecular characterization is somehow not that easy. Though I would say there are some institutes and laboratories which are now doing in India also the characterization
Right.
work, and they are doing it. And we hope in soon we can find a proper medication for COVID regarding our Ayurvedic, so much the whole thing we have for in our Ayurveda. So I wanted to ask that you have shown Tulsi and its globalization. Does the impact of different geological places have also on the different biochemicals or the secondary
Exactly.
metabolites
Yes,
in
yes.
Tulsi?
It is important. It's a very nice question. This
Yeah,
is something which we have studied, and you can find it also in our paper, which was published in 2018. So
yeah,
definitely yes,
uh,
because the plants evolved in a certain geographical location, and they
yeah,
developed some adaptogens, some
Hmm.
features. Like for example,
Hmm.
the volatiles of tulsi are stored in trichomes or glandular hairs. So for
Hmm.
example, if there is or the anthocyanin deposition in Krishna tulsi, because if you grow
Yeah.
Krishna
Hmm.
tulsi or
Hmm.
Sham tulsi, which because that's
Yeah.
what we call it, Sham or
Hmm.
Krishna tulsi. So if we
Hmm.
do if we grow Sham or Krishna tulsi in a shaded area, it will
Hmm.
not have that pigmentation, that anthocyanin deposition will not happen. But the
Yeah.
moment it goes in the sun and it gets
Yeah.
UV light, enough UV
Light.
light, it turns. And this is what
Uh,
we have tested here. So we have
yeah.
used external sources of providing UV, and
Hmm.
of definitely yes. So if I want to get tulsi ji from India,
Hmm.
tulsi ji is not the same which I grow in Baden-Württemberg of Germany, because the
Yeah.
Baden-Württemberg Germany, Ram and Krishna tulsi are both green. But
Hmm.
in India, I can, if it's Ram tulsi, it will anyhow be green, but the Krishna tulsi will turn purple or dark greenish in color. So definitely yes, the geographical location has an important role to play in this, in the volatile
Hello.
and in the chemical
Yeah.
profile.
Yeah. Okay. And one more question is that there's, as you said, that the flowering or the periodism do affect, as you said, in anthocyanin pigments, and it affects the flowering also. Like that also, I think biomolecules in different varieties are also, I would say, they get generated or they are synthesized according to the stress also. And
Correct. There's a lot.
stress,
Exactly.
stress,
Yes. So it
yeah.
depends on the environment. Like for example,
Yeah.
if you induce drought, for example,
Yeah.
there'll be a lot of hormonal changes in the plant. Like for example, the jasmonic acid pathway
Yeah.
is activated, and that leads to a lot of other pathways like phenylpropyl pathway, which leads to lignin deposition, etc., etc. So all these things add up to the chemical profile. So definitely yes, you are right, and this is like known, and this has been well studied. And also, I mean, if you go through some of the papers from
I wanted to ask you that checking the stress, do you take in the in vivo plants, or you do in the culture level?
no,
What
we do it in vivo plants. We do it
in
in vivo
vivo
plants.
plants? Okay.
We also have tulsi cell cultures, just
Okay.
in case
Yeah. Yeah. Cell
anyone is
lines. Yeah.
interested. We have cell
Yeah.
lines, and maybe next time I come to India, I can bring it for
Hmm.
D.D.U,
Yeah.
so people can work on it. So cell lines are easy to handle, of course, and cell lines
Yeah.
are you can do a lot of
Actually, they are
things on
easy to
cell lines.
maintain in the next
Exactly.
batch
Yeah,
culture,
yeah.
and we can say
So,
precisely when the something can be given, the proper acid
right,
treatment can
right,
be given.
right,
Right.
right.
So it was a great lecture,
Yeah,
sir. Yes,
Dr.
Binay
Chaube has
sir.
a question.
Yeah, yeah, yeah.
Um, it's a wonderful talk, first of all.
Thank you so
And
much.
I'm basically a virologist. I'm from Calcutta University. And the point is that you have shown about moringa and tulsi. So I
Yes.
have tried both the plants, and I tried several plants also for their antiviral efficacy. So have you tried them on RDRP acid?
No, no. Actually, we are a lab which is purely plant physiological lab. But
Uh,
if someone is interested, like for example, let me give you an example. There's this eugenol which is there in Ocimum species, and eugenol is kind of not. I mean, according to the European health standards, it's not certain. If you go beyond a certain level of eugenol intake, it can have side effects, or it can also lead to problems. So in I mean, this is something which we are working on, but we are working on more on the genetic level to understand the mechanism of eugenol production. So but we have yes, there's a good thing that we have not tried it, but we would love to. We are open for collaborations if someone is interested.
in my lab, I have used, I do culture HIV and hepatitis C virus. Okay.
Right.
And we have both cell culture based as well as in vitro based assays. And there
Mhm.
I have used this moringa. And
Uh-huh.
I have seen wonderful activity against HIV. And we are in the process of purifying the molecule. And
I,
so that might be in near future we will publish that. But this is one very important area.
I wanted to ask you a question. So did
Yes.
you try the extracts of leaves, or you just tried the whole
No,
plant or the fruit?
we have seen only the leaves so far. Only the
Okay,
leaves.
my suggestion is that this is just out of the literature which I have read. I mean, the traditional literature and the observations, because uh, uh, animals give us a lot of input. So please try the flowers. I mean, I'm really, because that's something which is lacking in literature. If you can do that, it will be wonderful.
Okay.
Okay.
We have tried with those you said saddle, that fruit of it,
The fruit.
but we didn't get much activity in that. But flowers we got, and unfortunately, actually in Calcutta city, we don't have too many wild these moringa plants growing around here. So that's a problem. But we will certainly try. Whenever the flowering is there, I will try to manage to get some flowers. And and we have tried it against hepatitis C virus also. And there also we have seen. So my suggestion is that this plant is having a significant RNA polymerase inhibitory activity.
It is correct. Yes, I have seen literature which talk about this, which is important for antiviral
Yeah, and this,
mechanisms.
I think this can be, and if somebody is in the group, somebody is growing or culturing, say, COVID, or they can try this plant against
Right.
COVID also, because that is also having a RNA RT with them. So
Right.
wonderful.
Right.
And wonderful.
Thank you. Thank you, Dr.
Binod
Chubey.
sir. Thank you, Binod sir. Thank you, sir. So one more question is there that that moringa also has a lower glycemic index and it helps in the diabetic patient. So
I mean, yes, there is.
yeah,
It's. I mean,
yeah,
if you search literature, it's full of
yeah,
this
yeah.
reports. Yeah, that's true.
So
That's
so can I know have you tested some of the metabolites or you know
true. No. So as I said that we are not a nutritional lab, or we
yeah,
do
yeah,
not have the
yeah.
facility. So we are working mostly on, the authentication of the Ayurvedic products.
But the biochemicals you have taken that are from the leaves,
That's from the leaves.
leaves.
Yes, that's from the
Okay,
leaves. Yeah.
leaves only. Okay. So thank you, and you have definitely enlightened us and given us impetus to carry
I
good
mean,
work because I'm
no
as
no
a tissue
questions
culture.
from students. No questions from students.
The questions are from students right now. So because
I would like to take this opportunity also to thank Dr. B.N. Pandey from BARC for the lovely talk he gave. And in the beginning, I could not thank him for that. So thank you very much for
yeah,
that talk. It was really nice of you to give us an insight on this radio therapy,
yeah,
and
allowed to.
really nice.
Thank you very much,
Yeah.
Dr. Sahi. And since your surname is Sahi, actually I remember one of my friend was there, Mr. Sahi, when we were doing our BSc from Gorakhpur city. So after
Okay.
listening to your talk, I remember him also. So actually, this is we got a lot of connections. Actually, since Moringa, you talked actually in. We also work some of the. Since I know that we work in radiation biology, so in some of the work actually in. 2000, I work on eugenol, so radiation protection ability of eugenol. Already afterwards, so some literature is there, and actually moringa actually whether it can be tried for some of the radiation protection ability or cancer or radio sensitivity applications already it is known. I'm not very much aware about this literature.
No,
But that
no.
is some of the area.
What I can say is that there are reports of moringa having some effects against radioactivity by usage of moringa roots and moringa bark, but I do not
Bark,
know about the
yeah.
leaf. Because leaves and fruits and flowers are well studied, but the roots on the and the bark of moringa are not so well studied. So that's and being from bark, I mean, it shouldn't. You have more access to facilities, so it will definitely be a very good. And also because of this radioactive thing which you talked about, moringa is known for phytoremediation. So that's something where one can connect the phytoremediation aspect with radioactivity, but I'm not sure about it. I mean, I'm talking just based on
Yeah.
some speculative literature which I have
Since,
read.
Literature, yeah.
yeah, since discussion arising, and now we have got botanists sitting there, and now you got virologist and you are a nutrition person. So one thing maybe I suggest that in Department of Botany, or maybe what happens that many times why we are not able to catch up this research, that many times we are not developing these plant molecules rational based. Like what happens that suppose you take a plant, you take a root, you take flower, you take fruit, or you take bark, all the different components have different ingredients. So not necessarily that all ingredient will work for different applications. Like suppose, for example, if I'm going to look for radiation protection, I may have a different requirement of molecules. If I have to look for some radiation station, I have to look for a different types of molecule. So when we are going to look for applications, we can go for more rational based research rather than taking some fraction and looking for, and ultimately we are finding that they are not working or they are not really useful. So when we are doing this type of research and I feel that Department of Botany, they should try to take up this type of research more, where they can, instead of doing like if most of those who work on extracts, they take either plant, they take root or shoot, but they don't have proper rationale that okay, why they are taking root for this application. So many times we are not able to achieve that what we are ultimately looking for it. So this is one of my suggestions since this all discussion came. So this is like I thought I just share. And since we work on radiation biology, so our main application is more radiation protection or radiation sensitization or decarburization of some heavy metals or radionuclides. So these are our main focus. But certainly I appreciate the research Dr. Sahi doing actually for this type of applications. This is one of my just comment.
Thank you, sir. Thank you.
Yeah.
Thank you, Dr. Sahi. And also I thank Dr. Badrinaran Pandey also for giving such insight. And actually, as a tissue culturist, I have also done work, and as a student only, I was listening, not as a faculty member to you. And
we are all students actually. We are all students. I feel.
Yes.
Science.
Yeah.
Student of science.
I was listening, and I was getting the insight where to start a new thing. And I would definitely like to say that we are all have been enlightened, and thank you for joining.
Thank you for the chance to give a chance to me to talk. So thanks to all of you.
Yeah.
Thank you very much.
Thank you. And once again, I hand over the mic to Dr. Deepa.
Thank you, Dr. Tulika, and thank you, Dr. Shaili. Now I request Professor A.K. Dwivedi, sir, to give his message to the students. Professor Dwivedi.
Good evening. Am I audible, Deepa ji?
Yes, yes.
Okay, thank you, sir. Thank you, sir. So good evening, myself, Anil, and I'm very much delighted to learn the findings of two eminent scientists, Dr. Badrinaran Pandey, sir, and Dr. Sai, sir. I was just listening to their lecture very thoroughly. They have gone to their subject deep insight. And just as they are working on the recent fields, Dr. Badrinaran Pandey ji, he focused on the use of radiotherapy for the COVID. It is an alarming thing, and it is the demand of the time. We can come to some better solutions. We are very much hopeful to see something good from Badrinaran Pandey sir very soon. Thank you very much, Badrinaran Pandey sir. Then again, I am thankful to Vidyut Pratap Sahi sir. He also enlightened on the traditional topics, particularly the usage of moringa and basil, tulsi, the two very important and culturally ancient plants of our history. These two plants have been used ever since their knowledge, and he has done a lot. He has enlightened us through the literature of Vedic and all. So we are very much thankful. And on this platform, I also convey my regard to Dr. Vinay Chaubey sir, who made the discussion very interactive and enlightening all of us in this direction. And at this point, I also, as Dr. Deepa ji has assigned me the duty of giving the message, I have a very deep regard message for my patron, Vice Chancellor Professor V.K. Singh sir. I have deep sense of gratitude to our Dean of Science, Professor Subirnath Tiwari sir. Similarly, to our head, Professor V.N. Pandey sir, Without the cooperation and help of these dignitaries, this special event would not have been made possible for today. And also, I am very much thankful to all the faculty members of our department, and at the same time, the students or the scholars who are main beneficiary of these events. They are also acknowledged. I am also hereby thanking to Professor Pooja Singh ji, Coordinator Department of Microbiology, who coordinated this event very successfully. Thanks to all. Thank you very much. Back to
Thank
Dr.
you.
Thank you.
Thank you, sir. Now I request Professor Pooja ma'am to please give vote of thanks. Dr. Pooja.
Hello, everybody. I am Professor Pooja Singh. Sorry for not sharing my video because it will eat my bandwidth and I won't be able to talk to you. So I was listening through both the lectures. I enjoyed it a lot. And thank you, Pandey sir from BARC, and thank you, Shai ji from Germany. Please stay safe, stay
Thank
happy,
you.
you
Thank
have
you.
a very good smiling face. And I thank everybody who participated here and gave a new dimension in COVID treatment. I learned a lot from Pandeyji regarding the radiation therapy and how he has used his knowledge to combating in this COVID. And obviously, I also work on essential oils. I have been working from past 20 years in essential oils. So I was able to relate more self with Shai ji's lecture. Thank you so much for the insight in Moringa and to Osimum. I take this opportunity to invite both the scientists, please, whenever after this pandemic, please come and visit us and please share your interest with us. We will learn a lot from you. I take this opportunity in future. Please come and visit us. I thank everybody to be a part of this. And I have deep sense of gratitude to our Vice Chancellor, sir, our HOD, and Deepa ji especially, and she has become an expert in arranging webinars. Thank you so much for giving me this opportunity and for listening this webinar, and everybody who is being participating here. Thank you so much for being with us, and we shall meet again in some other webinar, and better we shall meet again in person. Thank you so much.
Thank you. Thank you so much.
Thank you very much.
Thank
you, all of you.
Thanks a lot.
Thank you.
Thank you, ma'am. And by this, we will end this session. So we thank you all, each and everyone who is present here. Now let us stand for national anthem.
Started.
Please start, Deepa ji.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता
पंजाब
अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा हिमाचल
जमुना गंगा उच्छल जलधि तरंगा
शुभ नामे जागे तब शुभ आशीष आए तब जय
गाथा
नायक जय हे भारत भाग्य विधाता जय हे जय हे जय
Thank you. Now we end this session. Thank you to all.